Okay, thanks. Okay, Sabah khair, everybody. Now, in this lecture, so we've covered antibodies and we've talked about how the generation of variability amongst all the antibodies that the immune system makes. We've talked about the different isotypes, namely IgM, IgG, and so on. Now we'll start off with the T cells, and we'll begin by describing how T cells recognize antigen, because even though they are recognizing the same antigens as antibodies, if we're talking about proteins, there's a difference in what aspects of the antigen are recognized, because T cells need an extra condition compared to antibodies. So basically, the objectives of this lecture is to introduce cell-mediated immunity as brought about by T lymphocytes, to talk about the differences in the mechanism of recognition and the targets between what an antibody recognizes and what a T cell recognizes. And the difference lies in the fact that T cells need to recognize antigen presented by these surface structures called MHC antigens. And we recognize the groups of lymphocytes and their function based on their expression of these surface molecules, which we call CD antigens. And then I'll say something about the T cell receptor. And then if there's time, I'll talk about how antigens are processed by things like macrophages and dendritic cells to load onto these MHC molecules to be recognized by these T lymphocytes. So first of all, this is what I was referring to in the introduction, that an antibody molecule, because it is bivalent, doesn't recognize a whole antigen, say in this case, a bacterial cell. It only recognizes proteins, although this is a generalization because antibodies can also recognize long chain sugar molecules like polysaccharides and lipopolysaccharides. But generally speaking, these so-called T-cell dependent antibodies recognize fragments of a protein molecule, what we call epitopes. These epitopes might be around 20 amino acids long. And if you recall your basic biochemistry, the amino acid sequence within a protein chain determines how that protein is folded because of the characteristics of each amino acid with regard to its electrostatic charges, its side groups, and everything. So these epitopes recognized by an antibody molecule can form different shapes, again, dependent on the amino acid. And these shapes may be linear or conformational, so that it may look like a rock or a spike or whatever. And if you had to denature this protein by boiling it or by treating it with chemicals or whatever, then you would change the conformation of that antigen such that because the shape is then altered, it would no longer be recognized by an antibody. So basically, an antibody has to recognize a protein antigen in its native conformation. T cells, however, recognize these small fragments, these epitopes, but the condition is that these epitopes need to be presented on these surface structures called MHC molecules, which are expressed on these specialized cells, which are taking up the antigen and then chopping them up into individual epitopes and presenting them on MHC molecules. Only in that way can the T cell receptor recognize the antigen. So the T cells need to recognize antigen in what we say the context of MHC. So just some general descriptions of cell-mediated immunity. So within the adaptive immune system, we divide it into two arms, what we call the humoral immunity, which is antibodies and B lymphocytes, and cell-mediated immunity, of which we divide into two arms, T helper cells and cytotoxic T cells. And within this classification, all T cells we recognize through their expression of this surface molecule called CD3. And within the CD3 group, we have T helper cells which express CD4 and cytotoxic T cells which express CD8. Within the CD4 group, these T helper cells need to recognize antigen in the context of class 2 MHC, and cytotoxic T cells recognize antigen in the context of class 1 MHC. So first of all, what are CD antigens? So every cell that's involved in the immune system, whether you're talking about macrophages, NK cells, neutrophils, express a wide range of surface molecules. These could be receptors, adhesion molecules, cytokine receptors, and so on. There are some of these surface molecules which are only expressed in cells of particular lineages. So for instance, CD14 is a toll-like, it's an accessory molecule to one of the toll-like receptors, TLR4, which recognizes LPS on gram-negative bacteria. So we use tagged antibodies against, say, a collection of cells and any macrophages there. These tagged antibodies are going to bind to CD14, and in that way we can count the number of macrophages there are in our collection of blood cells. Similarly, CD19 is a molecule which is expressed only or predominantly on B cells. So again, by tagging a collection of cells with antibodies to B19, we can quantify how many cells there are, how many antibody-producing B cells there are. The other markers are CD16 for natural killer cells, and then CD3 for T lymphocytes. So in the old days, when I was a graduate student, I would have to sit through the night under a UV microscope, counting the number of stained cells in a blood collection, and then basically quantifying how many T cells there were in the sample of blood that I had. Nowadays, with the advent of laser technology, a standard instrument in most immunology labs is this instrument called a flow cytometer. Basically, what it is it's an automated UV microscope. But instead of having to visually count every cell, this thing can do it automatically using computers. Basically, what it involves is that when you start off with your experiment and you have this collection of blood cells, you will then incubate those cells with a number of specific antibodies to the different cell types you're looking at. Say you use anti-CD14 to look for macrophages, anti-CD16 to look for B cells, anti-CD3 for T cells. Each antibody is tagged with a different chemical that will give a different color if that chemical tag is hit with a certain wavelength. You then pass this collection of antibody-stained cells through this reservoir, and this directs every individual cell to pass through this sheath of fluid. And as these cells come down, there are laser beams hitting into this stream of liquid, such that every cell which is tagged with a particular antibody of a particular tag will flash a signal. And these signals are then picked up by the computer, and you basically get a readout like this. So we look at this graph here. What we have is on the x-axis, we have the number of CD8 stained cells, and on the y-axis, the number of CD4 stained cells. So if you remember from your basic high school maths about reading graphs, we see that the scale goes from 0 up to 10,000 this way and this way, right? So in this lower left quadrant, basically these are all your negatively stained cells. These are all your red blood cells, neutrophils, macrophages, everything which is non-CD4 or CD8 positive. If you look along the x-axis, at the extreme end of the axis, these represent the highest stained cells tagged with the CD8 marker. So this represents all your cytotoxic T cells. Similarly, the top left-hand quadrant represents all your CD4 or T helper cells. And because the computer, every dot represents a cell. And so because the computer can give you a numerical readout of the number of dots within each quadrant, you can monitor the actual amount of T helper cells or cytotoxic T cells you have in the blood of patients during the course of a disease. So as many of you know, AIDS is caused by the HIV virus infecting CD4 cells. So with someone who's been infected with HIV and maybe develops AIDS and then gets antiretroviral treatment, we monitor their blood samples for the number of CD4 cells every few weeks to see if the drug treatment's working. So just to repeat, all T cells are recognized by expression of CD3, and T helper cells are CD3 positive and CD4 positive and recognize antigen in the context of class 2 MHC, and CD8 cells recognize antigen in the context of class 1 MHC. So now we say, what are MHC molecules? And MHC stands for major histocompatibility complex. So this refers to the surface antigens on all living organisms. So mice, rabbits, dogs, cats, humans, we all express MHC. We use MHC interchangeably in the case of humans, where we also use the term HLA. This is historical because there were two arms of research before they stumbled on what HLA or MHC was. The one was pure immunologists doing tissue grafting, where they would graft bits of skin from one strain of mice onto another and see whether that skin graft took or not. And then they determined that the conditions for engraftment or rejection depended on these surface molecules, which they called MHC. In the human system, surgeons were, after wars, were busy experimenting with transplanting kidneys, livers, hearts into different patients, and they came up with a set of surface molecules which they called HLA, human leukocyte antigen. Subsequently, it turned out that both groups were working on the same system. So if we talk, so for the human system, I will use the term MHC or HLA interchangeably, but just bear in mind I'm talking about the same thing. So in the human system, well, in all organisms, there are two classes of MHC molecules. Class 1, which in humans consists of three groups, HLA A, B, and C. And then class 2, which in humans are DQ, DR, and DW. First of all, class 1 MHC molecules are expressed on all nucleated cells in our body. So basically, these molecules act as barcodes for our immune system to recognize what is self versus non-self. So essentially, if you were to do a kidney transplant from taking a random donor who has a totally different HLA component to yours, you will reject that kidney very quickly unless you're on immunosuppressive drugs. So your immune system would immediately see that grafted organ as being non-self because of these differences in HLA. There'd be less, the rejection wouldn't be as fast if you received a organ from someone who is related to you. And you wouldn't reject it at all if you received the organ from your twin brother or sister, right? So the closer the HLA match, the less chance there is of rejection. There's so the HLA genes in all of the cells in our bodies are one of the biggest gene sets within our chromosomes. And this reflects all the generations that made us, all the different matches between males and females of HLA genes and recombinations that we inherit. What makes the HLAs different to other genes in the blueprints in our body is that the alleles that we inherit from both our mothers and our father are expressed. So what this means is that we have three groups of class 1 MHC, A, B, and C. That means that we inherit the A, B, and C alleles from both our mothers and our fathers, which means that we express a total of 6 class 1 MHC genes. All right? So I said that class 1 is expressed on all nucleated cells. Class 2s are expressed on specialized cells in the immune system, predominantly in what are called professional antigen-presenting cells. These are macrophages, dendritic cells, and B lymphocytes. So these three groups of cells will express 12 MHC molecules because they express 6 class 1 and 6 of these class 2 MHCs, whereas all non-immune cells only express class 1 MHC. All right? You look puzzled? So all parenchymal cells, the cells that make up our epithelial tissue, whatever, only express class 1 MHC. And within this group, we have 2 copies of class 1 proteins that we inherit from our mother on the one arm of the chromosome, and that we inherit from the father on the other arm of the chromosome. So both are expressed. In other words, they're co-dominantly expressed, right? So because it's 3 class 1 from the mother, 3 class 1 from the father, that's a total of 6 class 1 proteins. These antigen-presenting cells, not only do they express the class 1 proteins, so that's 6, but they also express the class 2 proteins, another 6. So that means that these 3 groups, dendritic cells, macrophages, and B cells, express a total of 12 MHCs, 6 class 1 and 6 class 2s. Okay? Then the other thing to note is that these class, whether it's class 1 or class 2, they always have tagged on their structures an antigenic epitope. So in other words, the MHC protein cannot assemble or be expressed on a cell without having an antigenic epitope sitting within the structure. So an antigen is part of the MHC molecule, right? So if we look at the fine structure of an HLA molecule, this is class 1, where it's essentially composed of one protein. And so this is the transmembrane motif, which anchors the HLA into the cell membrane. It's divided into 3, based on amino acid sequence, into 3 groups called the alpha 1, alpha 2, and alpha 3 motifs. Now, if you look at alpha 1 and alpha 2, that looks like Mickey Mouse ears or a hot dog bun or an air cushion, right? looking from this direction. And with a class 2, it's composed of two separate proteins, an alpha chain and a beta chain. But again, if you look at the beta 1 section, that is also a hot dog bun, right? The class 1 has this accessory molecule called a beta 2 microglobulin, which is a common plasma protein. And it sits there to stabilize this class 1 complex. Now, this hot dog bun, or this bed, is where antigenic peptide sits. So in this diagram, we're looking this way. So our hot dog or our antigenic peptide is lying longitudinally. This diagram changes 90 degrees. So this is our antigenic peptide. Remember I said that peptides or epitopes are composed of amino acids. Each amino acid differs with regard to the side chains it has. So depending on what amino acid is within this peptide will determine whether this epitope or peptide can sit or fit within this groove. So this is where these MHC molecules can only accommodate a particular hot dog sausage having the appropriate amino acid sequence. All right. And correspondingly, the T cell receptor, based on its structure, must also accommodate the correct amino acid region of the peptide, but also has to accommodate various structures on the MHC to be able to give you a close fit. So a T cell receptor needs to recognize not only the antigenic epitope, but also various structures on the MHC presenting that antigenic epitope. So this is why we say that CD4 cells recognize antigen in the context of class 2 MHC, because those antigens are only presented on these class 2 molecules, and CD8 recognized in the context of class 1 MHC. These peptides are around 10 to 15 amino acids long. Now, I said that an HLA or MHC molecule can only assemble if it has an antigenic peptide loaded within this groove. So you can understand that in the case of a virus-infected cell, these HLA molecules pull out these virus proteins and then express them on class 1 MHC, which is what the antigen recognized by cytotoxic T cells to come and kill these cells to prevent them from making more viruses. But under normal conditions in a healthy body, what is the source of these antigenic peptides? Bearing in mind that our normal cells aren't infected with any pathogens. So what is the source of these antigenic peptides? The source comes from the fact that all of the molecules that we make in our bodies, especially proteins, have a certain use-by date, have a certain half-life, so they degrade. So every minute, our bodies are breaking down the various proteins that make up all parts of our body into peptides. And so within within these cells, all the cells in our bodies, we are loading our class 1 MHC with these broken down recycled peptides. Our immune system is able to see these peptides as being part of our normal structures and then leaving those cells alone. So basically, these self-peptides are giving a don't touch me signal to the immune system. But as soon as you get an altered peptide loaded on this MHC in the form of a virus protein, in the form of a mutated protein, or a tumor, a cell that's starting to develop into a tumor cell, presenting altered peptides, that will signal to these cytotoxic T cells that there's something wrong here, let's kill the cell. All right? So every MHC, so these, all our cells are expressing thousands of these MHC molecules. And each MHC molecule can be presenting a different peptide. But the rule is that each MHC molecule can only express one peptide. It can't express multiple peptides. So what T cells do is, and predominantly T cells, they sniff every cell in our body, looking at these peptides on the MHCs. And if they see that it is self, they will leave it alone and go on to the next cell, right? Now, what about class 2 MHC? So the class 1 is predominantly refers to parenchymal cells, the cells that make up our livers, our kidneys, our skin, and so on. These so-called professional antigen presenting cells are the cells like dendritic cells, macrophages, and B cells, which grab outside extraneous pathogens like bacteria, viruses, fungi, and present them on class 2 MHC, which is seen by T helper cells. Okay? Some other types of cells, like parenchymal cells, well, like I said, all cells in the body express class 1 MHC. But some other kinds of cells, like endothelial cells, some epithelial cells in the skin, occasionally can also express class 2 MHC. This is just to show the recognition of antigenic peptide presented on, in this case, class 2 MHC by the antigen presenting cell. So this is the T cell receptor, which is seeing that peptide. And you can see that this being a T helper cell, it also has this additional receptor, CD4, which is recognizing a certain structure outside of the MHC molecule. So this is where the class 2 MHC restriction comes from as well for the T helper cell. Not only that, this pan T cell marker, CD3, plays an important role in T cells because the T cell receptor, just like a surface antibody molecule, is essentially just a recognition structure. It doesn't have an intracytoplasmic sequence for transducing phosphorylation signals to the cell nucleus. That is generated by the CD3 complex, so that as soon as the T cell receptor sees antigen, this CD3 complex joins onto the T cell receptor and then undergoes these chemical phosphorylations to transduce the signal to the nucleus to direct that T cell to make cytokines or be cytotoxic or whatever. So in other words, if this T lymphocyte didn't have a CD3 component and only had a T cell receptor, it would only bind to antigen, but then do nothing, all right? This is another representation. So I told you a similar thing happens with the B cell receptor, that whether it's a B cell receptor, antibody, or a T cell receptor, these are just for seeing antigen. They don't have this intraplasmic sequence. And so that function is brought about by the Ig alpha, Ig beta for the B cell receptor, and the CD3 complex, which is composed of 6 different subunits for generating these phosphorylation signals. A unusual reaction that you get with certain bacteria is that they produce these so-called superantigens as part of their virulence factors. These bacterial proteins can bind to MHC linked onto a T cell receptor without having a specific antigen there. So basically what the superantigen does is it activates the T cell receptor non-specifically. And so when it does that, it causes particularly T helper cells. This only happens with T helper cells. So the superantigen only binds to the class 2 MHC T cell receptor complex. And when it does that, it creates what's called a cytokine storm, because it super induces that T helper cell to make masses and masses of inflammatory cytokines. And these get into the bloodstream, and so then they can cause blood clotting and total organ failure. This is similar to what, it's not a superantigen, but this is similar to what COVID-19 is doing as well. So now let's talk about antigen presentation. So I said that MHC molecules cannot assemble unless they have antigenic epitope loaded onto those sausage binding regions. Now, just based on your basic cell biology, where does protein synthesis occur? In what part of the cell? The endoplasmic reticulum. So the ER is where MHC molecules are made. So the messenger RNA coding for class 1 or class 2 MHCs go into the ER, the ribosomes then make the proteins. And so both class 1 and class 2 are loaded or are created in the ER. Now, if we talk about class 2 MHC loading first, recall in the first lecture when I talked about phagocytosis, where we have phagocytic cells, and in this case, a macrophage or a dendritic cell. It phagocytoses an extracellular pathogen like a bacterial cell, takes it up into a phagosome, And within the cytosol, you have these lysosomes which fuse with the phagosome. And these lysosomal granules, like myeloperoxidase, superoxide, I think Dr. Brass covered that with you already in pathology, or not? Well, he will anyway. So these potent enzymes all function to degrade these bacteria cell into individual peptides, right? While that is going on, you have newly made class 2 MHC molecules being created within the cisternae of the ER. So here, this is the messenger RNA, these are the ribosomes, and these are the newly created MHCs for both class 1 and class 2. And as they are assembled, you have another protein called the invariant chain. And what this invariant chain does, it sits within the peptide binding groove, which allows for the newly made alpha and beta chains to come together, right? So think of this invariant chain like bubble wrap on something you just bought from Amazon, right? Once this newly made class 2 MHC complex is created, it then buds into the Golgi apparatus and then moves within the cytosol in this vacuole to a phagolysosome. Here, another protein called HLA-DM. Now, this is not a classical surface molecule like class 2 MHC, but it's actually an enzyme. What HLA-DM does is it cuts off this bubble wrap and selects an appropriate antigenic peptide to load onto to fit within the peptide binding group. And so then this is then expressed on the surface where it's recognized by a T helper cell. So the upshot is that the antigens processed in the exogenous pathway for class 2 presentation come from outside the cell through phagocytosis, right? For class 1 presentation, these are antigens which are made inside a cell, like virus-infected proteins, like bacterial cells which might survive and replicate inside a bacteria, inside a macrophage or a dendritic cell, or like normal cell peptides which are broken down, right? So let's say it's a virus-infected cell. So as the virus gets into the cell and starts to make its own proteins, remember that every cell has an internal immune system through the recognition of these PAMPs. These pattern recognition receptors within the cell can see that these proteins that are being made don't belong to the cell, right? So when that happens, the cell can then direct these virus proteins for degradation. And so it tags these foreign proteins with this so-called chaperone, ubiquitin, which passes the viral proteins through this cylinder called a proteosome. These proteosomes consist of multiple proteolytic enzymes. And what they do is they degrade this foreign protein into individual peptides. And these peptides are then carried into the ER through these two important gateway proteins called transporter-associated proteins, or TAP. Once they get into the ER, another protein which has a similar function as HLA-DM, called tapesin, selects for the appropriate peptide to load onto the class 1 MHC, and then that is moves into a Golgi apparatus, into a vacuole, and then expressed on the surface to be seen by a cytotoxic T cell. So class 1 loaded peptides come from inside, they're endogenous, right? And also, this is a function of that bubble wrap, that invariant chain. Because while this is going on, you're also getting class 2 MHC molecules being made in the ER. So that invariant chain prevents any antigenic peptides which were destined to be loaded on class 1 to be loaded onto class 2, because the newly made class 2 molecules are being blocked from loading for class 1 with that invariant chain. This is an EM picture showing these proteosomes. So as you can see, it looks like a cylinder. So as these proteins destined for degradation are passed through, they're then chopped up into the individual peptides. And during an inflammatory response, when you get cytokines stimulating macrophages and dendritic cells, it induces the cells to make more of these proteosomes. The upshot of this is that you are creating more antigen presentation on these antigen presenting cells during an inflammatory response, an infection or whatever, for presentation to these reactive T cells. Okay, I'll leave it for that and carry on next week.
We have a question, sir, in the chat. You want me to ask?
Yeah, let's get them.
Are you sure? So one of them, she was just a little bit confused about the MHC. So she's asking in regards to autoimmunity, saying the MHC1 receptors are recognized as non-self and pathogenic, right?
Yeah.
Okay. So the second part is, so if that's the case, then why are only some tissues targeted and not all? Because they express the same MHC1, right?
That's a good question. I will deal with that later when I talk about tolerance and autoimmunity. But that's a very good question. But just bear that in mind. But we'll come to that in a later stage.
Okay. And there's another one. It's called, she's saying, Doctor, can you please re-explain the variant chain that's related to the MHC?
The variant chain that's related to the MHC I don't understand what variant chain, because so variant chain only, if you are by variant chain, if you're referring to VDJ, right? I talked about this in the B cell, in the antibody. Remember, actually, this is a very good question. Remember when I talked about immunogenetics, and I said that the generation of antibody diversity is at the level of somatic rearrangement. So in other words, in a developing B cell, to make messenger RNA for a particular antibody, it selects one particular V exon joins it to a D exon and certain J exon, right? Similarly, T cells also use VDJ, but the alpha chain, just like the light chain of an antibody, only selects V and J, and the beta chain of a T cell receptor uses VDJ. So the T cell receptor, which is also by the two separate proteins, but the alpha chain is similar to the light chain of an antibody. But both the T cell receptor and the B cell receptor, the diversity in antigen recognition follows the same principle of this selecting out particular V's to join onto D's and J's and so on. Okay.
All right, and there's another one. He's asking what is the difference between cytotoxic T cell and natural killer cell in terms of what they recognize.
Okay, so again, I will talk about this later, but the upshot is cytotoxic T cells, which are lymphocytes, recognize, let's say in the case of a virus infection, virus peptides loaded onto class 1 MHCs. So if you have, say in the case of COVID-19, you've got a bronchial cell that is infected with the SARS virus and it's expressing the SARS peptides on the cell. If you have any antigen-specific T cells there, it will recognize those SARS peptides and kill that cell, right? So these are CTLs, cytotoxic T cells. Now you have some viruses like cytomegalovirus, which is a herpes virus, which escapes cytotoxic T cell killing because what they can do is block expression or activity of this protein TAP, and so prevent cytomegalovirus peptides being loaded onto class 1 MHC. And so basically what this virus can do is to hide from the cytotoxic T cells. But in doing that, they also prevent class 1 MHC expression on these infected cells. Now, natural killer cells need to see normal MHC class 1s on cells as a negative, don't kill me type reaction. So if they see missing class 1 MHCs, they will kill the cell. So even though a cytotoxic T cell cannot kill that cell because there's no MHC, a natural killer cell as a backup will come and kill it because there is no MHC. Okay?
All right, so we have two questions, two more. One is in organ transplant, do we look at all 6 HLA?
No, because with immunosuppressive drugs now, the stringency for HLA matching isn't as great. So we basically look at 2 class 1s, A and B, and one of the class 2s, EQ.
Perfect. And the last one is, can you please explain invariant chain?
So the invariant chain is basically a pre-packaging molecule, because remember that first of all, the class 2 is composed of 2 subunits, alpha and beta. And you don't get these 2 subunits grouping together without something occupying the peptide group. So the invariant chain functions as a surrogate antigenic peptide by occupying this peptide groove, allowing for the two subunits to group together. But an additional function is that while this is happening, class 1, newly made class 1 molecules are being loaded with antigen. So by occupying this groove, you're preventing these newly made class 2s being loaded with potential class 1 antigenic peptides. You're blocking those sites. Okay?
All right, perfect. Thank you so much.
Okay.
Right. That's it. I can,
You can do that.
I'll do it. Thank you.